on developing new breeding methods. He has co-released 64 cultivars and 36 germplasm lines or populations. Dr. Baenziger is an active in Group Science Society of America and has been a division chair of president, and as well is an associate editor and editor-in-chief of Group Science. In 2013, he received the Genetics and Plant Breeding Award from National Council of Commercial Plant Breeders. In 2015, he received the Lifetime Achievement Award from the National Association of Plant Breeders. He has more than 300 publications and has received millions of dollars in grants to support his research. Dr. Baenziger's presentation title is Reflections on Hybrid Winter Wheat Lessons Learned. So we are happy to have you, and yeah, please go ahead and.
Okay, well, thank you first of all, and thank you for the kind invitation to give this talk. It's a great honor to be part of this symposium. It's fun to follow my friend and colleague Amir Ibrahim. It's always good. We worked together very closely for many, many years. And so what I'd like to talk about is a little bit, if you look at the title slide, every author in red is from the University of Nebraska. Every author in maroon is from Texas A&M. Boha, pardon me, from Basnetz from CIMMYT, Joachim Riefs from IPK in Germany. Friedrich Longen is from University of Hohenheim in Germany. And then Jean Benoit, Serzan, and Thierry Moitié are with the Zhao plant breeding, which have been extraordinarily helpful in this overall project. Let me see if I can figure out how to, there we go. So the topics I want to talk about today are the lessons we've learned by working in hybrid wheat. I'm going to give you a little background on hybrid wheat. Then I want to give you an idea of why now. Why are the hybrid wheat programs restarted? Talk a little bit about our efforts, of course, our main collaborators with Texas A&M, Amir and Jackie, on finding and building Heterosis. We have sort of three stages. The first one was a diallel stage. The second one was the balanced missing crossing block stage. And now we're into the building Heterosis stage. And then we're going to end like Amir did, talking a little bit about how do you make seed economically. And I want to give you just a brief concept of what happens if we fail and what happens if we succeed. So the very short history of small grains, or of hybrid small grains and hybrid wheat. I started breeding in 1976. That time there were extremely active programs in the 1970s and 80s in the USA, in Europe, in Australia, the Republic of South Africa, China, and India. But by the 90s, almost all of the public and private programs had ended. Now, the efforts in Europe persisted. That would be Satin Union Research, which later became Azure Plant Breeding. And in 2010, and thereafter, many companies started working again on hybrid wheat. And so what changed and what stayed the same? Well, the first thing is, why did hybrid wheat fail in the 1990s? And one of the things I want you to understand is that there were very successful hybrids in the 1990s, okay? Cargill and Hybrid-Tech and a number of other companies had released very good hybrids. The difficulty was, and this is something Amir hit on, there were relatively few hybrid programs, but there were numerous and very well supported public and private cultivar programs. So the hybrids never really separated themselves from the cultivar programs. I'm going to come back to why that was important a little later in the talk. But they had an advantage, but they never could keep the advantage. And another problem was there was no sustained public hybrid wheat breeding effort to support the private industry. Carl Lucken at North Dakota State University, when he retired, all of the program ended. Now, the sad part was that the private companies were fragmented, they were working independently, and they constantly duplicated each other's research. So once again, there was an inefficiency there. And to me, it's a missed opportunity. Compared to hybrid rice, where there's now somewhere between 15 and 20 million hectares, there's probably 100 to 200,000 hectares of hybrid wheat. And why is that? It's the lack of sustained efforts, and it's the lack of collaboration. Now, what did not change was the yield. And I don't know if you can see my pointer, but I hope you can. If you're on the right side of this slide and you look at the developed world, you can see the productivity of wheat has gone to virtually zero, while hybrid corn is slightly above 2%. If you go to the developing world, which to me is on the left side of this slide, you can see that rice and wheat are 1% per annum, and corn is a little bit over 2%. So as Amir said, we're going to need 1.4 to 1.7, maybe higher. Right now, we don't have mechanisms of getting to that level of productivity. And that's something which we desperately need. Now, the greatest changes for hybrid wheat is that we now have the hybridizing agents that allow you to make and test experimental hybrids. That was huge. Second part is our inexpensive next generation sequencing, which allows us to develop dense maps on our parent lines. That then led to excellent genomic predictions and also the possibility for hybrids where we can test a subset and really predict a whole large number. That leads us into the era of big data. And also that leads us into finding and building heterosis. Every crop that has heterosis found it and then built upon it. And we have a much better understanding of the CMS system and fertility restoration. So the first slide is, I just wanted to show you, this is a slide of a 3-year supply of Quasar 100. It's the hybridizing chemical that we use from Azure Plant Breeding. They have been outstanding to work with. It's not perfect, but it's very good. It's a contact plant growth regulator. We have used, in general, single spray dates, and I'm going to explain why we got that. That's possible for us, but at the same time, we may go to multiple spray dates, but it's a very effective compound. First year we had it was 2015. We thought 7 seeds or less would be 75% sterility. We were underneath that the first year. From 2006 on, we're more like 90% sterility in most lines. Historically, there will be some lines that are difficult to sterilize. That's normal. There is a chemical interaction with the genotype, but basically we can now make hybrids. Okay. When you make the hybrids, what you should understand is the chemical hybridizing agent is a plant growth regulator. Anytime you make a hybrid, you first of all sterilize the seed. The gape occurs usually about 3 days after the pollination should have occurred, so you have a shorter grain filling period. If there's any phytotoxicity, you will have shriveled seed, which we have here. You also see some shriveling in the CMS because the gape is delayed there also. And that's compared to a normal self-seed. The interesting part is that if you work with Quasar 100, okay, you'll discover that the shriveled seed has absolutely no effect on the yield of the next generation. And we did that because we made hybrids that would be the equivalent of the self-seed, basically crossing line A with itself, make a hybrid. It was shriveled, planted the plumped normal seed, and there were no differences. It does have some funny effects. If you're dropping shriveled seed through a planter, it tends to flow through a little more slowly, so you see a little more stagger in your plots, but that's minor. We found that reciprocal differences were rare, and that was huge because that means you can make your hybrids based on the biology, that you don't have to pick one side to be the female, one side to be the male. You can do whichever one gives you the best synchrony between the floral pollination times. In Nebraska, our window of pollination is relatively short, so usually one application works. And as I mentioned earlier, there is a genotype by chemical by environment interaction on inducing sterility, and that Azure Plant Breeding has been great to work with. I want to come back and show you when I say that generally one application will work. And if you look at that, this is a study by Nick Garst. It was using 299 lines. The lines came all the way from Texas to Montana. They represent the full width of germplasm in the Great Plains. And we looked at how different were the pollination dates, which would be the pollen 50. And in some years, you might get as much as 10 to 15 days, but in other years, it was only 4 or 6 days. And so that tells you that if you're that tight on your pollination dates, you probably only need a single application. Again, as Amir was saying, pollination date is highly heritable. Everything that's early stays early. Everything that's late, which is in green, stayed late. That's also true for anther extrusion. It's true for height. We did look at some pollen duration with hoping we could find lines that diverged more frequently or gave you a longer window to pollinate. That only occurred in non-stress years where you had a sort of a more normal pollination. Stress years, everything was tight around just 2 or 3 days. So looking at flowering date, and this is the elite trial, 60 entries, and we have it from 2016 to 2021. And we look at the earliest and the latest flowering. You can see that on average, it's somewhere between 4 and 8 days. In an odd year, it might be as many as 13. If you look at the regional trials, which would be lines again from Texas all the way, in this case, to Canada, further than Montana, and you look at the range from earliest to latest, they can be as late as 15, but most commonly they're 6 to 8. So that just gives you an idea of why in Nebraska everything shrinks to a sort of a common flowering date. The optimal stage for using the hybridizing agent is when the immature spike is 12 to 20 millimeters. That's the one that can be seen on the right. One that's too early is one that is on the left. That's more like 8 millimeters. The staging does not take into account the head length differences, so we may want to adjust for that in the future. The biggest concern you have when using a hybridizing agent is late tillers because they're at a different developmental stage. So we plant the females at very high rates of seed, so you have very few tillers. We plant the males at very low rates, so you have lots of tillers and hope to expand that tillering capability and the pollination window. Again, the key is excellent anthers for the male and excellent gape for the females. Now, when we worked on the crossing block, and this is work by Hannah Stahl, we started wanting to see how did gape relate to yield? And then we decided we should correct for how did gape with a good male relate to yield. So if we looked at the correlations for yield adjusted to the gape and then adjusted for the parent, the male parent, we found very high correlations, much better correlations. And it simply says to have a hybrid wheat, you're going to need a good female and a good male. Hannah's conclusions were that for us, the gape angle at 100%, which we had pretty good heritability on. This is all visual scoring. She was very, very careful. And in our environments, it seems to work pretty well. The female traits seem to be more heritable than the male traits. The visual scoring was very easy to use. And we actually think genetically there may be a simpler explanation of the female traits than there is for the male traits. Male traits are always highly heritable, but very difficult to find underlying genes. The big question we have is her data was on hybridizing agent-induced gape, and will it reflect the CMS-induced gape? And we're going to come back to that a little bit later. All of that was done with our dialleles, or beginning with the hybrid wheat crossing blocks. We then started using the German designs by Joachim Rief and Friedrich Longen, which is a balanced, incomplete, missing design. And the basis of this is if you have really good markers on your parents, which you get with your genotype by sequencing, you can test a small group of hybrids and predict a much larger. The example that we used is that with Texas and ourselves, we developed a crossing block of 50 males and 100 females. Half the males, half the females came from Nebraska, and the other half came from Texas. In theory, if you have 150 lines, you can make 11,175 hybrids. That's far too many for any of us to make, far too many of any of us to test. But we were able to create 700 hybrids. And the way we did that is we took 25 males and we crossed those males to 14 females. Now the females changed with each male. so that basically all 100 females were represented when crossed to the 25 males. Then we took a different set of 25 males, the other to get to 50, crossed them again by the 14 females, and that gave us one location in Texas, one location in Nebraska. The idea being if we had a catastrophic year and we lost one location, we still would have at least hybrids produced somewhere. We could test 700 hybrids, and by the genomic predictions, we could predict the remaining 10,475 hybrids. And once you have that level of hybrid testing or predictions, you can start building your heterotic pools. And again, even if we lost one crossing block, we still could predict 7,750 hybrids, again, showing the power of what predictions can do. Just a quick look at the lines. Everything in red was from Nebraska. Everything in blue was from Texas A&M. The lines often clustered by their breeding program, but because we share germplasm, you'll see some clusters included lines from both programs. We did find, using our augmented designs to test this was quite difficult, that there was a trend, a negative trend between kinship and hybrid yield, which basically says as the lines become more closely related, you tend to lose a little bit of your yield. That's not a surprise. We also tended to look at the additive genetic contributions, and this is from the diallel study, and it was predominantly based on additive genetic variance. And this is important because some other programs have disagreed with this result, which is okay, that's not a surprise. But the other thing that happened out of that was that it explains why Anneal's work in Amir's group, that the F2s do predict the F1s. Additive genetic invariance suggests that. Our study corroborates what Anneal already found. Now, if you look at genomic predictions, and this is a very critical spot, and I'm going to take just a moment to explain this slide. Now, this is using the diallel experiment. 325 hybrids, full diallel, so we made reciprocal, so we tested 650, but because the reciprocal differences were rare, we functionally had 2 replications, which means we had a very good estimate of phenotypic variation. These on the x-axis are, you have 650 data points, you remove 10 at the NA10, and then you use the 90% you have remaining to predict that 10%. The one that's more interesting to us is the NA90. That says that you have your 650 data points, you know what the yield is, you remove all but 10%, 65, and you use the 65 to estimate the remaining, which is like 585, okay? The reason why that's so important is if you look at the German algorithms, they're saying if we make 700, we can predict 10,000. You need to be able to use a very small subset to produce and predict a much larger subset. We have all the data, so we're able to look at our predictions. And yes, as you use fewer lines to predict more, they go down, but they're still remarkably good at the predictions. We then went to the balanced missing and complete factorial design. The blue and green bars are from the Texas sites. They have a little better phenotyping capabilities than we did, better heritabilities. The Nebraska's in the more salmon colored. But once again, you can use 10% to predict a much larger pool and still do it reasonably well. That's the take-home message. And that led to this table here. This is 11,175 hybrids, all predicted from the 700 we tested. Anything in red has very good yield. Anything in blue has relatively low yield. And so now we've been able to predict a massive set of hybrids by testing a relatively few. Now, the question is how well does that algorithm work? So we looked at the 11,000 hybrids. We made 700. There were lots of hybrids that we were predicted should do very well that we never made. So we started making the hybrids that were predicted well. And in this experiment, which was a 3-replicate experiment, 230 hybrids, 25 parental lines, that's so we could do some mid-parental estimations, Heterosis, 10 common checks. The 10 checks are common to every study in Texas and Nebraska, so we can always tie them together. 55 hybrids, okay, were better or equal to the best pure line check, which is this line in yellow at the very bottom. Now, I want to compare that to what we had done previously without being able to predict. So we made random hybrids. We're curious to see how well did they do relative to the best commercial line available. And in using the 6 environments that we had in Nebraska, 0, 7, 9, 10, or 18 out of 600 lines. So we're looking at, less than 3%. We're better than the best commercial check. Yet in this case, 24% were best. We did have one location where 200 hybrids beat the best commercial check out of 600, and that's because under stress, hybrids are always far more climate resilient. Okay. The best commercial heterosis was 11%, averaged over 3 environments, which is a much more robust test than in individual locations. But that line is a Texas line that's not widely grown within Nebraska. It had a great year. If you looked at the lines that would be more commonly grown in Nebraska, the commercial heterosis was 19%, or almost 900 kilograms per hectare. Heterosis has to be found, it has to be developed. And that's what these prediction models are telling us. And there's also G by E for heterosis, as you would well expect. Now, if you look at measuring heterosis, and Amir gave a great history of that, so I'm not going to spend much time on that. But I wanted to answer a question that often comes up: how much heterosis is needed? Ideally, a grower should receive $2 in benefits for every dollar they spend. So if he spends $1, he expects to get $3 back, one for the dollar he spent and $2 in benefits. If you could get seed costs to $20 per unit and you plant 2.5 units per hectare, which is roughly what we'd use in the Great Plains, that would be $50 in seed costs. The increased value should be $150 per hectare. Now, when you think about heterosis and the value, it's based on 2 things: grain yield and the value of the crop. So as the value of wheat goes up, the level of heterosis can go down. And that's very important. It's much different if you're having, say, $7 a unit wheat versus $3 a unit wheat. Now, hybrids are more risk avoidance, more risk tolerance, especially stress tolerance. So that also is a factor you can come in. Now, we always present heterosis as percentages, but that's always deceptive. 20% heterosis on a 2 metric ton per hectare crop's only 400 kilograms. 5% heterosis on a 10 metric ton per hectare crop is 500 kilograms. Grower would much rather get a 500 kilogram benefit than a 400 kilogram, even though the heterosis is actually much higher in the lower yielding sites. Now, this leads to one other thing that when you work with hybrids that people sometimes don't remember, and that is if you have excellent parents, you need a lower level of heterosis. If your parent lines are 106% of the best commercial check, then if heterosis is 9%, the hybrid will be effectively 116% commercial heterosis. 1.06 times 1.09 is about 115.5. If your parents are only as good as the best commercial check, then the heterosis has to be 16% to get to 116% of the best commercial check. A lot of people think that heterosis is relatively fixed, and it's the parent lines in the background that are really helping on the hybrids. So, and we talked about the previous failure of hybrid wheat. It may be that the background, the best commercial checks just keep increasing so rapidly, they never could build a hybrid program fast enough to stay ahead. You do have a concern, excuse me, that farmers could save F2 seed. The prediction is the heterosis will be reduced in half. That's fine, okay? And that would be much higher than the best commercial check. The way to get around that is if you use CMS, so you have to have cross-pollination. That will reduce the yield of the F2. Or if people don't like the lack of uniformity in the segregating of the F2. So if we recap, we've used the balanced incomplete factorial designs. We've started developing our heterotic pools. Early insights, this is where the pools are. These are the Nebraska pools. There are 10 lines in each pool. We did that so that we would be broad enough to not have inbreeding depression, but narrow enough we could manage it. As you look in Nebraska, as you would expect, most of the lines are from Nebraska, but there's a very good selection from Texas. Now that we have the pools, we can also begin to develop testers. Two very good commercially released lines, Ruth and Rubidoo, both with good pollination characteristics, look like they could be the testers to help separate out future lines into which pool they belong into. We did the same thing for the blacklands and the bushlands, the two major ecozones in Texas. They have their lines. Now they're a little bit earlier than ours, so they tend to have fewer Nebraska represented, much more Texas. Interestingly, if you look at it, the Nebraska pool of 20 lines, the blackland pool of 20 lines, and the bushland pool of 20 lines, there are some lines that are in multiple pools, and in fact, 2 lines are in every major regional heterotic pool, which we think is must be extremely good combiners. Now, our early thoughts are we're going to use our 10 lines to develop the recurrent selection programs for better parent lines, but we're also going to be going back to our traditional cultivar program using all of our predictions and genomic selection and annealing algorithms to develop new inbreds, which will go right back into the crossing blocks, right back into the yield trials, and can be separated into pools. So we're going to link the cultivar development with the hybrid yield trials. Our future studies, we have intermated every line from the A pool to all the lines in the B pool, the idea to better understand heterosis. We are intermating lines from within the A pool and the B pool, and that's to begin to develop the pools so you can start getting the crosses that you need to build the populations, okay? And as we've mentioned, we've now begun to develop testers. We can now relatively quickly more broadly test lines outside of the pool. It helps that Nebraska wheats are generally later than most wheats, so that they're outstanding pollinators, and they would be difficult if you had them as the female because they would nick even later. Now, Amir talked a lot about the commercial hybrid system, the 3-line hybrid system, and the only point I want to mention here is twofold. One is that the A by B line is the difficult cross because they're identical except for the cytoplasm. And what you'll see is when a line pollinates, it doesn't gape. It expects to be pollinated, and when it doesn't get pollinated, then it gapes. So there's always a delay between a gaping line and when it would normally pollinate, which makes this synchrony very difficult. The hybrid wheat production, because you can pick late R lines to pollinate after the A line has gaped, is much easier to make actually. So I just wanted to give you some idea of how we did when we were looking at our CMS materials. You have lines here. The female was a CMS female. These are the yields of the males. These are just grams per plot. And we just wanted to get an idea of how good our cross-pollination was. Now we can tweak this and do a lot better than we've done it. But basically, without tweaking, your cross-pollination in the A line, B line series is about half. So that is by far the more difficult cross to make. And it's as low as a quarter. So it just shows you that to have a pollinator that's pollinating earlier than your line is opening up to receive pollen is a challenge. I also want to talk about the importance of seed purity. Impurity in any parent line is magnified in the hybrid. It's really difficult that way. In this set of lines, you can see what's in red is the female, the CMS female. It's completely surrounded by the male. Then we have a barley fill. This is in a barley field, so there's as little wheat pollen as we can possibly get in our fields. To be a highly cross-pollinated wheat should be 15 meters away from any other source of pollen other than the male that you want. That's why we put the barley fills in. That gets us, if you're going to do this commercially, you need 30 meters. What we have discovered is that it's extremely important to keep the females pure. So we're now going to mow out the front and back males so that when we run our combines up the middle row, we will only be harvesting the CMSC. We also are now going in and taping lines that are truly, or marking lines that we know are truly CMS hybrids, or CMS lines. and we're going to hand harvest those, and all that is to avoid combine contamination, any chance of getting fertiles in there. It's a very difficult type of thing. Now, an overlooked but critical cross, and this is one that I can't emphasize enough. If you look at this cross, the R line is the female and the male is the B line. The purpose of this cross is twofold, because when you look at the F1, it looks like a very traditional hybrid of an A line as a female by an R line as a male. And you don't want to go to the risk of producing A lines that will not perform in hybrids. So if you have a B line, that's what you start with, then you convert it to an A line, okay? This cross will tell you if there's any inhibitors of restoration. So if the B line has a gene which makes it hard to restore, you'll see that in this F1. If you allow the F1 to segregate, it will also tell you that your R line is in the right cytoplasm. Everybody wants it in the Timothy cytoplasm, so you know your restorers are working. And also, if you look at the segregation of fertile, partial fertiles, and steriles, you'll get an idea of how many genes you have, whether or not they're working, how tough your environment is. And that becomes extremely important because there are inhibitors of restoration, there are weak restorer genes, and this is the cross that lets you separate that out. Now, hybrid wheat is going to change how we look at the genetics of wheat. Virtually every study we do on genetics uses RILs or double haploids now, okay? We rarely are using heterozygous materials. We may look at the F1 for dominance, and then we go to inbreds. Well, if you have a fully sterile CMS line and a fully restored R line, but that's a homozygote. When you make the cross, the F1 will be heterozygous, and that's where you can see the strength of your restorer genes. And I think you're going to need to look at the heterozygosity of disease, insect resistance genes, all of these types of things, because you're seeing more of these types of questions, especially if hybrid wheat takes off. Last question, or one of the last questions, is the CHA phytotoxic. We know it is a plant growth regulator. The way to test that is actually to compare CMS lines to CMS lines that were sprayed with the CHA. The CMS line should be sterile, so there should be no effect of the CHA on inducing sterility if you have a partial sterile, and that the difference should be basically just to the chemical treatment. Now, this study is not perfect because when we did it, we discovered there were some impurities, which is why we've spent so much time cleaning them up, where there were fertiles in here. So the CMS here could have some fertiles which would aid in cross-pollination, and B, would also increase the yield. We used a common male Ruth, which is late. But the point is, I wanted to show you this, that you can see that the CHA may be slightly phytotoxic, but for some lines, there's a very good response to it, basically only a 20% yield reduction. Other lines, and this may be due to a prevalence of fertiles, you had a much lower ability to cross. So there could be phytotoxicity, but this is the way you look at phytotoxicity. Now, we've talked a lot about the three-line system and the CHA system and the like. The ideal system for hybrid wheat would be apomixis. And this is a paper in rice, and there's papers in wheat where you can see that there is efforts going forward on the how to make apomictic wheat. Now, this is very high-risk research. There were massive programs in the 1990s, and they all exited the business with the exception of Sutton Union, now with short-plant breeding. What if we fail? Well, there's two important papers. Strategies for selecting crosses using genomic prediction in 2 wheat breeding programs. And then another paper, Cross the Best with the Best and Select the Best, HELP, which stands for Hybrid Enhanced Line Performance in Breeding Self-Pollinated Crops. Basically, both papers say if you can estimate the F1 and then its progeny, and you can do that through molecular markers and the algorithms we've looked at, or in our case, you can actually make the F1 and test them, you will know exactly which crosses to make, which crosses to work with. So even if we fail, we should develop a much better and a much more robust cultivar development program. But what happens if we succeed? Well, first of all, there'll be less risk to the producer. due to climate change. That's extremely important. Hybrids are far more climate resilient than our pure lines. That's always been the case from all the data from the 70s, 80s, 90s, and up to now. You're going to have more productive food systems and better food security, which is what our goal has been. We will see wheat breeding change. In the US, it's roughly equal public-private venture to a largely private opportunity. If you look at the corn breeding efforts, they're predominantly privatized in the US. Competition will increase initially, and then it will probably reduce. There are only 3 major corn companies left in the US. They're very expensive, very profitable. The investment in wheat will increase, and that's not only for the genomics, genetics, breeding, but also for all of the other parts of a more valuable crop, more pesticides, more nutrient management, greater care of the environment, etc. And finally, the infrastructure for wheat will increase. If you look at the great seed companies, the Cortevas, which were formerly Pioneers, Bayers, which were formerly Monsanto, Syngenta, etc. They were all based on hybrids. And I think we're going to see the same thing. With that, I'd like to stop. I'd like to thank you and ask questions. Again, we'd like to acknowledge the research was supported by USDA competitive grants. We'd like to acknowledge all of our partners in this research. So with that, I'll stop, and Amir and I will both take questions.
Okay, thank you very much, Dr. Steve. And yeah, as I mentioned before, your presentation was like informative, and we are super lucky to have you. Well, so anybody has any question, please raise your hand, or you can write it down in the chat room. I can read it for you as well. Also, yeah, question for Dr. Amir, also question for him. Happy to answer it. Yeah, so Amir and Murad say like, thank you for this interesting talk. Glad to see you again, and yeah. So the question, yeah. So Amir and Murad asked a question. Did you get a chance to study some important traits such as disease resistance in the hybrid wheat program? I mean that there may become linkage between CMS and disease susceptibility and resistance. Just a thought. That's Amira Murad.
First of all, if I can answer this one, it's a pleasure to have Amira ask that question.
Yeah.
Okay, and I did mean to acknowledge we also have linkages to Massoud University with great students who would come here to work, and also excellent visiting scholars. And Amira is one of the great students, as was Ahmed, as was Shams. Right now, we don't believe there is a disease and CMS relationship. What Amira is talking about is that the hybrid corn industry was based on the Texas cytoplasm, and then there was a major disease, helminthosporium, which has now got a different name. I think it's bipolaris matus, that completely attacked the cytoplasmic male sterile system and ended that system. That's why all corn is now detasseled. And so the question always is that going to happen again? So far in wheat, it hasn't. It doesn't happen in every crop. Sorghum, like wheat, has a perfect flower, and it would be impossible to detassel. It's using a cytoplasmic male sterile system, and there hasn't been a disease that has attacked the sorghum CMS. But it's always a question, and it's a great question to ask.
Yeah, so thank you, Steve. Is there any other questions? By the way, you can send directly your question to our great presenters through via emails, and emails actually in the conference book. And yeah, okay, I think, thank you very much, Steve. Yeah, as mentioned before, we are lucky to have you. Also, Dr. Amir, we are super lucky to have you too. And yeah, thanks again.
Okay, bye, and thanks for the invitation.
You are welcome anytime, Steve.
Bye-bye.
Okay, bye-bye. So, Dr. Ahmed Atif, Okay, so now our third and final presenter, Dr. Ahmed Atif Salam. He is Associate Professor of Genetic Department, Faculty of Agriculture, Asyut University. Dr. Ahmed actually earned his scientific degree, bachelor and master from Department of Genetics, actually, Asyut University in Egypt. His PhD from Division of Plant Breeding, University of Göttingen, Germany. He worked as a postdoc scientist for 2 years at University of Nebraska and Lincoln in USA. Dr. Salam received the PhD award from German Academic Exchange Service, DAD. He received a recognition of high scholarship and outstanding achievement from the Honor Society of Agriculture, Gamma Sigma Delta Foundation in USA. He received State Encouragement Award in Agricultural Science in Egypt. Dr. Salam published high-profile and highly impacted international journals, such as Scientific Reports, PMC Plant Biology, Frontiers in Plant Sciences, Frontiers in Genetics, and International Journal of Molecular Scientists. He also a variable reviewer in a lot of international journals, such as Journal of Advanced Research, Theoretical Applied Genetics, BLOS 1, Frontier and Plant Scientists, and many journals. His research interest is improving abiotic and biotic stress tolerance in cereals using QTL mapping, genome-wide association study, and genomic selection. Dr. Ahmed, we are happy, and personally, I have to thank you very much for your organizing this international symposium and coordinating. And we are supporting you, and I mean, your support is unlimited. And thank you. You are such an excellent professor, actually, and not only in Asyut University, I think in